Okay, today is the last part of studying gene structure. And what better way to end than with terminators? So, part three, how is transcription terminated, and how can we find terminators in our phage genomes? So, our goals for today's lecture and also lecture in class are for you to, one, review the structure of a gene, and be able to describe and illustrate the two types of bacterial transcription termination mechanisms. One being row-dependent termination, and the second mechanism, which we'll probably spend more time talking about, is row-independent termination. And that means that we don't need row for this termination mechanism. And another name for this, and we'll probably stick to this latter name, is intrinsic termination. We'd like you to be able to identify intrinsic terminator sequence elements in a mycobacteriophage genome or a Gordonia phage genome, and be able to predict likely locations of terminator sequences on a genome map. So let's review the gene structure. We've learned a lot about promoters, we've learned a lot about the open reading frame and Shine-Delgarno sequences, but so far between last semester and this semester, we've left termination of transcription as something that happens in this little gray box after the open reading frame. So let's talk about more what this structure is like and how it triggers termination of the transcription elongation complex. So there's two types of terminators in bacteria and phages. One is dependent upon a protein called Rho, and so it's called row-dependent termination. And I'm going to talk about this in more detail in a minute or two, but it essentially involves this protein called Rho, and it actually comes as a hexamer that forms around nascent RNA or newly synthesized RNA, and it binds to this specific sequence in the RNA called RUT, or row utilization element. And it translocates up the RNA sequence until it reaches a paused RNA polymerase, and it essentially disrupts the DNA-RNA hybrid between the newly synthesized RNA and the DNA within this elongation complex. And there's another protein here called NUS-G that enhances this process. The second type of termination that we'll talk about probably the most in class is row-independent terminators, and these do not need the protein Rho. And the reason why we'll talk about these more is because these are very easy to find in genomes, whereas identifying row-dependent terminators is very, very difficult. So in row-independent or intrinsic terminators, essentially termination occurs because of a specific sequence in the RNA sequence that allows formation of a hairpin, and this hairpin disrupts the elongation complex and terminates transcription. So let's talk about both of these methods in a little bit more detail. In order to understand termination, what ends transcription, we have to understand what makes transcription very stable. And the stability of the RNA polymerase interacting with the DNA is dependent upon the structure of this elongation complex. And so this brown part of the diagram are the beta prime and beta subunits of the RNA polymerase that Keith described a couple of weeks ago, and here are the two alpha subunits. And so this brown, the brown subunits form what looks like this clamshell, and there's channels in the RNA polymerase, and there's one channel called a secondary channel through which triphosphate RNA nucleotides come in and act as substrate for the RNA polymerase and are one by one added onto the RNA chain. And there's also an exit channel through which the newly synthesized RNA chain exits the complex. Now, what makes this a really stable complex is the interaction of the RNA polymerase with the DNA. So it interacts with about 18 base pairs of the unwound DNA helix. So this is part of the DNA helix that has not yet been opened up by the RNA polymerase, unwound, but it is interacting with it. And then here we see probably about 12 base pairs that form the transcription bubble. This is where the DNA helix has been unwound. Now, the interactions with the DNA stabilize this complex, but what's really important is this RNA-DNA hybrid that is absolutely key for the stability of this complex. So there should be about 9 or 10 base pairs here of the RNA that's base pairing with the template strand within the transcription bubble. So disrupting this RNA-DNA hybrid is what is going to be key for disrupting the complex and terminating transcription. So let's talk about how these two different methods of termination disrupt the RNA-DNA hybrid. So with row-dependent termination, we have our DNA strand, we have RNA polymerase, which initiates transcription. And once it initiates transcription successfully and is able to begin synthesizing RNA, eventually the RNA chain, if there's a sequence, and we're going to call this, let's get our little pen out, we're going to call this RUT, which for row utilization element. This sequence is recognized by the hexamer proteins, row. That was some poor animation there. Row will basically encircle the nascent RNA chain. And again, this has to be an RNA chain that is not blocked by ribosomes. And this hexamer is basically a translocase, and it has helicase activity as well. And in an ATP-dependent manner, it will travel along behind the RNA polymerase as the RNA polymerase continues to transcribe. But eventually, the polymerase will pause at some sort of sequence after the open reading frame. And when RNA polymerase pauses, this allows Rho to catch up and interact with the elongation complex. And what is absolutely known and for sure is that it disrupts the RNA-DNA hybrid and therefore disrupts the elongation complex. And there's a couple of different ideas on how it does this, but what's important for you to understand is that once it catches up, it does disrupt the RNA-DNA hybrid, and the elongation complex dissembles, and transcription is terminated. So with intrinsic or row-independent terminators, the termination is all dependent upon the RNA sequence itself, and the RNA sequence allows secondary structure to form that will terminate transcription. So let's talk about how this works. The red shows the DNA sequence that occurs after the open reading frame of a gene. And what's important about the sequence is that there's basically a palindrome here within the DNA sequence, such that once the RNA is synthesized, it forms a little stem here. Okay, so there's base pairing between these complementary bases within this palindrome. Now, following the stem immediately is a trail of poly U, or a trail of U's, and this is called an L-type terminator simply because the structure of the hairpin and the poly-U trail here forms an L. This is the type of terminator that is found in E. coli, and in order for termination to occur in E. coli, for intrinsic termination, it's dependent upon having both a hairpin and the poly-U trail in the RNA. Okay, so let's talk about how this works. With intrinsic terminators and the mechanism of termination, it's, again, dependent upon disruption of the RNA-DNA hybrid. So I'm going to review the elongation complex. This is a different diagram. Here we have the gray showing the beta prime and the beta subunits forming the clamshell around the DNA. We have about 18 base pairs of DNA that are interacting with the RNA polymerase and stabilizing the interaction. Here's the secondary channel, and here's the exit channel for the newly synthesized RNA. And notice that the RNA has this secondary structure. So as the RNA comes out, it's interacting and base pairing with itself in different ways. Here is the transcription bubble, and here are the 9 to 10 base pairs forming the DNA RNA hybrid. So what happens is the RNA polymerase essentially will pause eventually after transcribing the open reading frame, and this occurs at a particular sequence. And in this case, this occurs at the poly-U tract. And so the majority of this, or the entirety of this RNA-DNA hybrid is made up of A's in the template and U's in the newly synthesized RNA. And you guys know that A and U base pairing are weak base pairs compared to CG base pairing because there's only two hydrogen bonds between an A and a U. So a large track of A's and U's is very, very weak and unstable. Okay, so let's figure out how this poly-U trail and weak bonding between the RNA within the RNA-DNA hybrid allows termination. So here, just upstream of the hybrid is where the palindrome occurs, and what happens is that eventually this sequence allows a stem to form within the exit channel, and this is a very, very stable structure. And what it does is, as the hairpin forms, it essentially is ripping off the poly-U sequence from the poly-A sequence in the template. So it's shearing away the RNA-DNA hybrid and disrupting that hybrid, and therefore disrupting completely disrupting the elongation complex. And so that is one way it is thought that intrinsic terminators terminate transcription. There are a couple other theories on how formation of this hairpin disrupts the RNA-DNA hybrid. This one with hybrid shearing is the most favored theory on how the hairpin disrupts the hybrid complex, and so that's the one we're going to describe in class. So what I'd like you to do to prepare for class is to draw a model of an operon, or you could also take a screenshot of an operon from one of your phage genomes. And I'd like you to identify regions in the genome that you would expect to identify a terminator. And the other thing you might think about are some of the be able to draw some of the alternative structures for terminators that are a little bit different than the L formation that we talked about in this lecture.